Hello everyone, welcome back to Microbiome Solutions. Today we are going to see about the types of ELISA. So the types of ELISA are mainly direct ELISA, indirect ELISA, sandwich ELISA and competitive ELISA. So direct ELISA is used in the detection of antigen. So from the name itself, it is a direct type of ELISA testing where the antigen and antibody reaction in a biological sample is detected. So it is used in the detection of antigen. Remember, for the detection of antigen in the given biological sample. So the patient sample will have an antigen and a buffered solution of the antigen to be tested is added to that antibody. Okay, so you will have a antigen in your sample that will be added to a microtiter plate. In the last video, we have shown that what is a microtiter plate. So where it is given time to adhere to the plastic through the charge interaction, which is followed by the addition of sample with an antigen. So antibody linked to an enzyme conjugate is added later so that this primary antigen as well as the antibody interaction will be proper. So the antigen-antibody interaction will be conjugated by an enzyme conjugated. This follows the addition of a substrate. Substrate is the component which stabilizes the antigen-antibody reaction with an enzyme conjugate and produces colored end product. This colored end product can be detected by using ELISA detector. So before detecting the colored end product, we have to stop the reaction or else the reaction will start occurring and the color may vary. So you have to add 0.1 normal H2SO4 or HCl as a stop solution for the ELISA. As you can see in the picture, this is an antigen of interest where you have it in the patient sample, and this is a primary antibody conjugated with an enzyme. Okay, this is the primary antibody which is conjugated with an enzyme, and this is a substrate. Okay, so your antigen is bound with the antibody with the enzyme, and then the substrate is added. You will get a colored end product. Next is indirect ELISA. It is used for the detection of antibody in the given sample. So direct means detection of antigen in the patient sample and indirect means detection of antibody in the patient sample. So here the microtiter wells will be initially coated with antigen specific for antibody. As we are detecting the presence of antibody in the patient sample, the substance which is complementary to the antibody is an antigen. So that antigen will be coated in the microtiter well against the antibody to be detected. Say for example, if you want to detect dengue IgM antibody, your ELISA plate will be coated with the antigen particular to dengue IgM. Okay, then it is followed by the addition of sample. Now first when you see the microtiter well, it will be coated with the antigen, and when you add the sample as it contains the antibody, because we are going to detect the presence of an antibody, there will be antigen antibody complex formation. Okay, once this antigen antibody complex is formed, after washing, only the unbound antibodies or antigens will be washed off, and after washing you are adding the enzyme conjugated with secondary antibody. Okay, this enzyme conjugated secondary antibody will be an antibody to the primary antibody. Okay, so you're having an antibody already in the patient sample. So this enzyme conjugated secondary antibody will act as an antibody to the primary antibody. Okay, then which is then followed by a substrate. After the addition of substrate, as you know, which forms a colored end product. This is the diagram showing indirect ELISA. So your plate, consider this as your microtiter well, which is already coated with antigen, and your primary antibody will be your antibody which is in the patient sample. Then you are adding your secondary antibody conjugate with an enzyme in it. Okay, this secondary antibody conjugated with an enzyme will act as an antibody to this primary antibody, and then after this, then enzyme conjugate, after the addition of enzyme conjugate, substrate is added. Substrate will give you a colored end product. When it detects the reaction of antigen antibody complex and the enzyme conjugate, the colored end product is formed from the substrate. Next is sandwich ELISA. It is used for detecting an antigen in the given sample. Okay, remember, same like a direct ELISA, it is also used for detecting antigen in the patient sample. Here, microtiter wells are initially coated with monoclonal raised antibody, which means this antibody will have specificity towards one particular type of antigen. Okay, so it is also called as capture antibody. Okay, monoclonal raised antibody or capture antibody will be coated in the microtiter well against the antigen to be detected. Okay, then this will be followed by the addition of sample. As I've written here, it is used for the detection of antigen. Your sample will contain an antigen. If there is antigen in the sample, it will be detected and a detection antibody and an antigen antibody complex is formed. Okay, so if you are detecting an antigen using sandwich ELISA and if there is an antigen in your sample, which will be detected by a detection antibody. So to this antigen antibody complex, same like all other ELISA, enzyme conjugated secondary antibody and a chromogenic substrate is added. After the addition of substrate, there will be color formation, which is measured by using an ELISA reader. Okay, here the intensity of color generated is directly proportional to the amount of antigen. Okay, in sandwich ELISA or if it is in direct ELISA, the intensity of color generated is directly proportional to the amount of antigen, which means if you get more colored end product, which says that you have that much amount of antigen in your patient sample, or if you're detecting an antibody, the reaction would be or the result would be the intensity of color generated is directly proportional to the amount of antibody in the patient sample. So that much color, how much color is produced after the end of your reaction, that much amount of antibody is there in the patient sample. So you can see the picture of sandwich ELISA. Why it is called a sandwich ELISA? Because the antigen is sandwiched in between two antibodies. Okay, so here there is a capture antibody which is monoclonally raised and we are detecting the presence of antigen. Consider this as a microtiter well and the microtiter well is coated with a capture antibody or a monoclonally raised antibody and then you are adding your antigen. So once the antigen is added, there will be an antigen antibody complex and then to detect this antigen antibody complex, the next one, a detection antibody is added. Okay, the detection antibody is added and then later an enzyme conjugated with antibody, secondary antibody is added. So this enzyme conjugated antibody will be acting as an antibody to this detection antibody. So you can see the antigen is sandwiched in between two antibodies, that is why it is called as sandwich ELISA. And upon the addition of the substrate, it will produce a colored end product. So moving on to the applications of ELISA. ELISA can detect both antigen and antibody and it is a useful tool for determining serum antibody concentrations. It is also found application in the food industry in detecting potential food allergens such as milk, peanuts, walnuts, almonds and egg. So other uses of ELISA includes which can detect the presence of mycobacterium antibodies in case of tuberculosis, detection of hepatitis B markers in serum, detection of enterotoxin of Escherichia coli in feces, detection of human immunodeficiency virus antibodies in blood samples. And the advantages of ELISA as it is sensitive, assay equipments are widely available, no radiation hazard, reagents are cheap with a long shelf life. Quantitative and qualitative ELISA can be used on most type of biological samples such as plasma, serum, urine and other cell extract. The disadvantages of ELISA includes only monoclonal antibodies can be used as matched pairs, enzyme or substrate reaction is short term, hence color must be read as soon as possible, which is a greater disadvantage. Though we are adding stop solution at the end of the reaction, the color should be read as soon as possible. Monoclonal antibodies can cause more than polyclonal antibodies because it is specific to one type of antigen. So in conclusion, ELISA or enzyme-linked immunosorbent assay is a novel technique useful in detecting both qualitatively and quantitatively an antigen or antibody which is present in a given biological sample. And besides its disadvantages, the technique is being widely used in diagnostics and drug screening. Chromogenic detection method used in ELISA is convenient and sensitive for any assay that is hazard-free. This is about today's lecture. We will be discussing about competitive ELISA in another unit. If you have any queries, please put it in the comment section. Please do like, share and subscribe to our channel. Thank you.